And we're official, yeah.
Okay, so in terms
All right.
of antibiotic reactions,
Yes.
you mentioned today, like knowing the type of enzymes that do those. So like underneath like this definition, there's like a little table. Is he gonna? Did he ask you all that? Did he like mention that in our review? And I was like, what? What chapter 10? The very last page, very last box of chapter 10.
He might ask y'all that.
Like these pyruvate and carbox.
Oh, he's totally.
Yeah, so that's, yeah. I think Andrew mentioned that last time. Well, I know we didn't know the definition of antibiotic.
Yeah, I'd probably say know the enzymes that are in blue. I don't think you're going to need to know where, so like the tissue or the organism. I don't think you'll need to know that, but definitely the enzymes, the enzyme names, what they do.
Wait, sorry. The enzyme names, just look it up. Like, what do you mean?
So
I can read pyruvate carboxylase, like that it goes to the liver, the kidney, or that it's a pyruvate plus HCO3.
no, I would say that mainly that those enzymes are anaplerotic, and like that can create TCA cycle intermediates. So definitely knowing the names of those. I don't think you'll need to know. Like what they actually do, that reaction overall. But if you wanted to learn that, I would say sure, go for it.
It's the very last page.
138. Good question. Any other questions? Any other questions?
I really have one. Let me just find it.
Did y'all just come to listen? Yeah. Listen to me talk? That's fair. Well, lucky for y'all, I'm prepared for that, so we're okay. All right. Who can I send? I've got a mock exam for y'all. I figure I'm going to send it to y'all right now. So can one of y'all just give me an email address real quick, and then y'all can post it on your Facebook page. Y'all can all pull it up, and we can just go through that. Do y'all want to do that? Two.
Sure. Okay.
Someone give me an email address.
M. Irwick with an I, like crocodile. M. Parker.
When?
People spell it with an E. Or they say Irvin.
So it's IRW.
W-I-N.
Cool.
Yeah.
Is there answers? Okay.
There's answer choices, yeah. I have a separate one. Actually, let me see. I'm not going to give y'all the answers yet. I'll give y'all the answers later. So that way we can actually go through them without y'all cheating and looking at them first.
I like that. Thank you. But you'll give them eventually?
Maybe.
Oh.
All right, sharing is caring.
I found my question.
What's the question?
So in chapter 9, glycolysis, and it's talking about the regulation by the different like non-reversible enzymes.
Correct.
And for the PFK1,
Correct.
he's like, oh, I'm going to give you a critical thinking question regarding this. And it's PFK1 activity and fructose 1,6-biphosphate are reciprocally regulated.
And not
What does
just
that mean?
Are reciprocal.
Page 125. It's in the middle of the page, under PFK1.
So.
Regulated. He's like, I'm going to do a critical thinking question.
PFK1 and fructose 1,6-bisphosphate.
Yeah. Which PFK1?
So if we have
Fructose 1,6-phosphate, how's that
if you have a lot of fructose 1,6-biphosphate, then you don't need to make more PFK1.
yeah. So PFK1's job is to make fructose 1,6-bisphosphate. Yes? Yeah. So if we have a little bit of fructose 1,6-bisphosphate, we're going to need more PFK1 to make that, correct? To keep the reaction driving forward. But also if we have enough fructose 1,6-bisphosphate, we can say, hey, so we got a lot of this. We don't need PFK1 to say, hey, like let's make more. We've got enough. So if there's enough product, it kind of turns off the enzyme. But if there's not enough product, enzyme's going to turn on. Does that make sense?
So is it safe to say the one that's directly in front of it will be the one to turn it off?
Really anything after it can. Really anything after it can. But mainly the one right in front of it, yeah. Any other questions? Did you get that email?
No, not yet.
Cool. All right, we'll just start going through it. Word of mouth first. All right. What is the major tissue for fuel metabolism? Yeah. Tested in 2 days. We need more than 2 people saying it. Major tissue for fuel metabolism. Liver. Absolutely. Absolute liver. What is the common compound produced from, yeah, that's actually right. So, common compound produced from oxidative catabolism of proteins, carbs, and fats. Acetyl-CoA, right? That's the one that goes in between all of them. What cells secrete glucagon specifically? Alpha cells, right? So what cells secrete insulin? Beta, all right. And what tissue is that? Pancreas, right? So our pancreatic beta cells secrete insulin. Pancreatic alpha cells secrete glucagon. Glucagon, does that increase or decrease glucose in the blood? Increase. So that means insulin decreases, right?
Wait,
Good.
rephrase that, sorry. I just answered what you said.
So does glucagon increase or decrease blood glucose?
Okay, increase in insulin. So it's triggered whenever there's low.
Exactly. What are the 3 forms in order of insulin? Pre, pro, mature. So where is it? Pre-pro insulin, pro insulin, mature insulin. So, good. What is the major effect of insulin on blood glucose? We already did that one. All right, chapter 7. I'm just going in order like chapters, kind of just how it runs through until y'all can get this thing. What's the definition of Gibbs free energy? Usable energy, right? Yeah, that's really all it is. What is the second law of thermodynamics? Ooh, yeah. Is that the first one, or what's okay? So if that's number 2? What's the first one?
It's constant. Energy's constant.
First one is created or destroyed. Second one is
Something about heat.
entropy. It has to increase or
Second law of thermodynamics. The total entropy of a system must increase if the process is to occur spontaneously. All right, so rule number 1. First law, energy is constant. Energy cannot be neither created nor destroyed. It can change forms, so it can, it can be released as heat, or it can be captured and remade to do work, but it's never made. There's never more of it or less of it. It's always the same. It just changes form. So that's number 1. Number 2 is the entropy of a system must go up. The randomness or disorder of the system must go up. All right, so if I've got a KEQ of positive 3.7, what's a possible value of delta G? Positive, negative, or 0? Negative. Why do y'all say negative? Good. Yeah, so if we have a KEQ that's greater than 1, so it's kind of opposites, that means delta G's got to be less than 0. Right? So if we've got a negative delta G, if we've got a KEQ less than 1, delta G's got to be greater than 0. And then if KEQ is equal to 1, then it's got to equal 0.
So less than 1 is positive, more than 1 negative, 0, or 1 is 0.
Depends on what you're saying is positive or negative.
Like the G.
So if G is negative, then KEQ is greater than 1. If G is positive, KEQ is less than 1.
Is this on your computer?
Yes.
The overhead show it?
If I awkwardly hold it this whole time? Y'all want me to awkwardly hold it this whole time?
Is this what you're sending us?
Yeah.
Then no. Who's sending us this? So we can do it like on our own later. No, I like it.
I mean, I could do it.
Email it?
I emailed it.
Yeah, then just pull it up on.
How about I just do this? Oh my god.
Did you get it right? Did you give him the right email? Because some people give you the wrong email and they just send a really good email sometimes.
Just play better.
Work, bitch. I thought my email was S. Phillips plus S. D. Phillips, and Scott Phillips got my email.
All right, is this better?
No, my email.
Is that, y'all can see it? Cool. All right.
Who's Scott Phillips?
Good question.
Uh,
Go to school here.
all right, number 9. An endergonic reaction is irreversible and will virtually go to completion. True or false?
And it's exergonic. It's like this.
Why?
I just said it.
I don't hear a lot of confidence in any of these answers. That's why I keep asking. Good, yeah, so it's exergonic. So if an exergonic reaction is highly negative, that's irreversible, right? So those are going to be, what reactions for glycolysis? What numbers?
1, 3, and 10.
1, 3, 10, and for Krebs cycle?
1, 3, and 4.
1, 3, and 4, right? What are those enzymes for glycolysis?
Hexokinase, and then PFK and pyruvate kinase.
Yeah, so hexokinase, PFK1, and pyruvate kinase. And for Krebs cycle, those are going to be yeah, so citrate synthase, isocitrate dehydrogenase, and alpha-ketoglutarate dehydrogenase, right?
Yeah.
So why is it isocitrate dehydrogenase and alpha-ketoglutarate dehydrogenase? Why are we saying that those are irreversible?
No,
They release carbon dioxide.
Yeah, because they're releasing that carbon dioxide. So we've broken that carbon-carbon bond. We're doing that for a reason. We don't want to put that back on. So that's helping drive the reaction forward. But so the release of CO2 from both of those enzymes is what makes it irreversible. Good. Good. Yeah, so the release of CO2 is going to make that irreversible. So the reason I'm saying that NADH is not what's making it irreversible is because we have that malate dehydrogenase over on the other side, right? And that makes NADH, and that's reversible, correct? So it's the release of the CO2 which is causing it.
So that's exergonic.
Yeah, so this one was false, should be exergonic. Skip 10. Endergonic reactions are termed anabolism. True or false?
True.
True. What's the other term? Good. Number 11.
Catastrophe. Right?
Yeah.
Good.
Or it's catastrophic, if that's what you're thinking of. All right, number 11. Which of the following is not a nucleoside triphosphate that can also participate in a high-energy phosphate group transfer?
Oh, this is a chart. Oh, that we talked about.
This is the one I was like, if you don't look at it, you're just going to see a bunch of letters and be really confused.
What DTP is not. Yeah. What page is that on? B. DTP.
Good. Good. So how we know this is that we know we've got guanine, uracil, and cytosine, right? We don't have anything that starts with D, do we? So that's how we're going to be looking for that. Taste taking strategies.
Well, he literally just asked it like that, or you think he'll be like ATP plus UDP equal like something like that? Yeah, it's so hard.
I'm not sure. This is how I remember it for our test. It's in like this, but we'll see. All right, chapter 8. What is the most important carbohydrate? And why? I got a couple glucose. We have any for glycogen? Do we have any for guanine?
Glucose.
We shouldn't have any for guanine, right? Because that's not even a sugar.
It's right here, that's why.
Yeah, it's glucose, right? So that's like the first thing that our body uses for energy is glucose. It's so easy to use. We can just put it through glycolysis. We'll pump out 2 ATP. Good to go, right? Our brain uses entirely glucose. So that's kind of important if we want to keep living and thinking and learning stuff for biochemistry. So glucose, glycogen, the storage form. All right. So
So it'll be second important.
no, we'll just keep it. We'll just keep it at first important. Let's keep it at first important because I don't know where fructose would fall under that actually. So true or false? Oligosaccharides have more than 7 covalent linkages.
False.
Why?
6.
3 to 6.
Polysaccharides are greater than 7.
Exactly. And so we've got monosaccharides, just 1, disaccharides, 2, oligo, 3 to 6, poly, more than 7. Good. Which of the following is not a ketone?
A. Glucose.
What is glucose?
Aldolose, sorry.
Yeah, aldehyde, aldolose, whatever. So what was our rule for learning these?
The euloses are ketones.
Mhm.
The roses are aldoses.
Exactly. So if you see anything that's in eulose, so like ribulose, xylulose,
Oh, that's fructose.
those are the ketones. But if you see anything that just ends in OSE, like glucose, with the exception of fructose, glucose, ribose, xylose, those are going to be just your aldehydes. So just look at the suffixes, and they'll tell you a lot about what it is. True, false. Only L isomers are used to synthesize carbohydrates in humans. God, y'all are so smart. Y'all are so smart. What is the storage form of glucose in animals? Just said it. Glycogen. What about plants? God, y'all are so smart. Sugars can be attached by which of the following linkages? O and P combination of any of those.
A and B. O and N.
So D, is that what we're saying?
Yeah.
That's right.
What page is that?
What page is that? I used to have all the page numbers on here. It's on page 109.
So, what is the question? O and N?
So really it's just saying they can be attached by an oxygen or a nitrogen. Really.
One of the three, oxygen, glycogen, ones is like serine.
Where are you at?
What?
Tyrosine.
Where are you at?
Tyrol.
You like mentioned in class, but it says like under the O link it shows like serine, but it can also be
Serine.
tyrosine and threonine.
I don't have anything for that. But if you said it in class.
And then what was the 2 N ones?
N is any
Amine and amide.
or CONH.
I was still right. That's what he said today. I was sleeping.
What is it?
Amine and amide.
Wait, just repeat the whole question. No, the N link ones is it's ASM, which is it's ASM. So O.
N-linked is NH2 amino acids that contain NH2 or the CONH2.
I forget what ASN stands for. All right, so for the record, so we've got how many for O-linked and what are they? We've got 3 for O-linked. Serine, tyrosine, and threonine. And then for N-linked, we've got
Amine and amide.
amines and amide groups. Good. So I guess that was just written in y'all's notes for y'all.
So O was what is it? Serine, threonine, tyrosine.
All right, chapter 9. Which of the following is not a storage form of glucose? Give y'all a second to read over this one. Why are y'all saying galactose?
It's a sugar. I know it's starches.
All right, what is it? You don't know.
Glycogen, starch, and sucrose are all the storage forms of glucose.
We were right.
So it's galactose. It says like on the chart on page 114, it shows like glucose and things all going out of it, and at the top for storage it goes glycogen, starch, and sucrose.
Yep, so the answer is going to be galactose. It's on page 114. So glycogen, starch, and sucrose. So what makes up sucrose? Glucose and fructose, right? So that's a disaccharide. So it's probably going to be more storage, right? Because there's multiple monosaccharides. So galactose is a monosaccharide, right?
Yes.
Y'all thinking through that, or y'all just agreeing with me?
I'm just agreeing.
Good. Okay. Yeah. So answer on this one is D, galactose.
Because galactose and glucose.
Yep. So, true, false. Glycolysis takes place in the outer mitochondrial membrane. Why? Where does it take place? Cytosol. Gotta know it. Gotta know it. True, false. There's a net gain of 4 ATP from glycolysis. 2. Why? What is 4? Bingo. So net gain 2. Overall, we make 4, but because we use 2 in what steps? Which enzymes use ATP? Good. And those just happen to be the irreversible ones, right? The ones that use ATP are irreversible. Which of the following enzymes participate in irreversible reaction during glycolysis? Bingo. So y'all can see how these can just jump out at you if you really know it, like the back of your hand. It's just like, oh, it's just going to be a breeze. What is the enzyme in glycolysis that produces NADH?
I concur.
I can't pronounce it, but I can tell you it's glyceraldehyde 3-phosphate dehydrogenase.
And why are we saying it's D?
Dehydrogenase releases added hydrogen.
Yeah, so if we see something that says dehydrogenase, we're taking off a hydrogen, probably putting it somewhere, and in this case, it's going to be NADH. Exactly. So what does PFK1 make? What's the product? What's the product that PFK1 makes? Fructose 1,6-bisphosphate, right? So then, what does aldolase make?
Makes the glyceraldehyde and the ketone of the aldehyde.
Yeah. So that's the next enzyme after PFK1. So we've got the fructose 1,6-bisphosphate. We've got the 2 phosphate groups on there. That's too big of a word. We need to simplify it, right? So we're going to use the simple aldolase just to do that. We're going to make glyceraldehyde 3-phosphate and dihydroxyacetone phosphate. Yes?
Mm-hmm.
Okay. And what does phosphohexosaisomerase do?
It makes it nicer. It turns glucose into fructose. No, it turns the glucose 6 into the fructose 6. So I said glucose.
Phosphohexosaisomerase. Yeah, it turns glucose 6-phosphate into fructose 6-phosphate. Sorry, I just had to think about that for a second. Good. So answer is D. What are the enzymes that are responsible for the production of ATP in glycolysis?
Pyruvate kinase, so A, and the phosphoglycerate kinase, D. A and D.
I was reading 24 and I was really.
You all agree?
Yeah, we do.
All right. Why not B?
Because that's the
cycle.
There you go. So understanding which enzymes are in which cycle is going to be really important. So he could easily just throw one of those in there to confuse you. So read the question in entirety. Make sure if you're understanding where you're at. I think he might go in order. Do you know if he kind of went in order with the exam last test?
Different forms, so some people would, some people wouldn't.
Okay.
Sometimes you get
So he may not be like this nice and just kind of go through order with y'all. So just be aware of where you're at and which cycle. 1,3-bisphosphoglycerate is formed by what enzyme?
Oh, that's the trio.
No.
It's the phosphoglycerate kinase. Or,
No.
It's the second half of it. Was second half of glycolysis.
It's not
glyceraldehyde 3-phosphate dehydrogenase, which is on there.
No.
It's phosphoglycerate kinase.
It's kinase. Yeah. then you go to 3-phosphate
Yeah, no, it's
glycerate, and then 2-phosphate.
It's formed from what enzyme? So the one that makes 1,3 is glyceraldehyde 3-phosphate dehydrogenase.
Scroll up just a second so it shows on.
Is that better? Sorry.
Step 6, we're showing.
So we're on number 24.
You're right.
It's not on there.
Yeah,
It's not on there.
You sure? Oh,
Glyceraldehyde
Yes.
3-phosphate dehydrogenase.
yeah.
So you go from glyceraldehyde.
just making sure y'all are paying attention. That's good.
This phosphate.
So I still haven't gotten that email.
Seriously? We gave the wrong email? What happened? Maybe spelled it wrong.
Oh, delivery has failed. It's awkward.
All that.
I sent it to.
Megan.
All right, so number 25. What cofactor is necessary for the reaction catalyzed by a hexokinase? Magnesium, right? All day, every day. Is there any other cofactor in glycolysis that we use?
And there's potassium for the last step.
Which one uses zinc? Do we know?
Always. If, yeah, if it's in bacteria, then it uses
What she said.
Aldolase.
Do we know which step?
I concur. Yeah, good. Zinc is used potentially in aldolase. I didn't even know that, so good. Well, y'all are killing it. Y'all are so smart. Y'all are so smart.
Brittany's smart and just said in the episode.
True, false. Glucokinase is present in all tissues, and hexokinase is present in the liver.
False. It's hexokinase type 4.
No, it's opposite.
No.
It's opposite. It's opposite.
It's false.
Hexokinase is in all the tissues, glucokinase is in the liver.
And glucokinase is also hexokinase type 4. Yep.
Yeah.
National Board's TQ.
National board question. Yeah, so would it make sense that glucokinase, so to go through glycolysis with only glucokinase as opposed to the more general hexokinase? So we want the general hexokinase is present everywhere, right? It's everywhere. But specifically, glucokinase is only present in where?
The liver.
In the liver. So it's specific, it's more of a specific enzyme, so it's got a specific spot, whereas hexokinase is more general, just talking about a 6-carbon sugar. So that's everywhere.
What did you add to that? You said TQ for hexokinase.
How many FADH2 are produced from glycolysis?
Zero.
0. How many NADH?
2.
2. Gosh, so smart.
You taught us well.
What molecule inhibits the action of hexokinase?
ATP?
ADP.
It takes an ATP to
Well, if you have ATP.
Yeah, you don't need ATP.
no, it costs an ATP.
Cost an ATP.
But you have to have ATP in order to.
Spend the ATP to make ATP, so if you have too much, then you don't need
This is a trick question.
No, but if you have ADP, it's not going to happen because it won't donate the phosphate.
You have to use ATP to.
That's what I'm saying.
And ATP is coming in.
No, that's what I'm saying. It's inhibiting. It's ADP. Because if you have ATP,
Okay, yeah.
it's
I
not
thought you said it was
inhibiting.
ATP. I agree. ADP.
ADP.
So y'all's final answer is what?
ADP.
ADP. I think the answer is ATP.
Is that because you finished the whole thing and then you're like, I have enough energy for now. I don't need to start it.
I'm pretty sure it's like, hey, so if we have a lot of ATP, do we need to go through glycolysis?
No.
Well, then it should say large amounts of ATP, not just ATP. If you have just one ATP, it's
Y'all wanted tricky wording questions, so I gave y'all tricky wording questions.
So that's talking about the entire state, or that's talking about the entire process of glycolysis. Then, not that's the context you're looking at, correct?
Well, in general, hexokinase. So yes, hexokinase needs ATP to be able to convert glucose into glucose 6-phosphate. Yeah, but that's on like the micro scale. So large scale, if we have a lot of ATP in our body, we don't even need hexokinase to work in the first place. There's really no signal that says, hey, hexokinase, turn on, because ADP is going to tell hexokinase specifically to say, hey, we need ATP, and then it's going to use an ATP. It's going to invest that one to make another one. So this is like a large scale picture. Does that make sense?
Yep.
Is that confusing for anybody?
So one more time, what did you say the answer was?
ATP with the,
The one
like
it
Tom.
says in the book is glucose 6-phosphate, which is kind of makes sense. And then for PFK is inhibited by ATP, and so is pyruvate kinase. Where are you at?
Like,
Page
but the
125.
plus two. So if you're not as short as ATP, there's no reason to even begin the whole process.
Yeah.
exactly.
So, yeah. Makes sense.
Good? Cool. And this is just my wording. I'm not like, that's just what I think. So I mean, we can ask, y'all can ask him tomorrow in class if that's right or not. But I think it's kind of right.
Yeah, no, like on a macro scale, that makes sense.
So cool. All right. What has the ability to inhibit the function of pyruvate kinase?
ATP. That's one in
I agree.
Yeah, it's C.
C.
Why would it not be A?
That's the one right above it.
Because yeah, that's what forms it.
It is reversible, so maybe if it like just went
I guess. Yeah, well, it would be ATP 'cause it's the same as the one before.
All right, yeah, so it's really what comes after it is what it inhibits, right? That feedback inhibition, so like you produce it and then that comes back and turns it off. Remember he draws that like A, B, C, D, E comes all the way back and turns off A.
It could also be inhibited by acetyl-CoA and long-chain fatty acids.
So it would be this right here. Yeah.
It says
From-
in the book it could also be inhibited by acetyl-CoA and long-chain fatty acids.
What page?
Page 125, the whole regulation by...
Yeah, so page 125, I would just definitely make sure y'all look over those. This is kind of where I'm getting all these from. He really likes to know what regulates, what doesn't regulate. So that's kind of where I'm going with this. So why not magnesium? Why would magnesium, why wouldn't that inhibit? Yeah, it's just a cofactor, right? It's just probably going to help it, if anything. Good. What type of reaction is phosphofructokinase 1?
phosphorylation.
Why phosphorylation?
Because you're adding a phosphate.
Say again?
Every time there's kinase, there's a phosphorus.
Exactly. Well, why wouldn't it be dephosphorylation all the way down here at the bottom?
You're
Because
adding.
it has one, it's adding a phosphate.
Exactly. You're adding it, right? So you're not taking it away. Great. What happens to pyruvate when it's fermented in yeast?
Ethanol. Because yeast for beer.
Yeah.
D.
D.
Sounds good? All right, chapter 10, last chapter. How many NADH are produced by a citric acid cycle?
3.
Good. What is the enzyme that produces acetyl-CoA?
A. A.
Yep. A.
We've got 3 A's. We have any others? It's definitely A. Got to know this name. Got to know this name. Got to know this name. Pyruvate dehydrogenase complex. We're taking pyruvate. We're turning it into what?
Acetyl-CoA.
Acetyl-CoA. All right. So we're dehydrogenating pyruvate. So we're taking off a hydrogen from pyruvate. I think actually a CO2 as well. and making acetyl-CoA. So it's describing the first thing and not the second thing. What's the only enzyme that describes the thing it makes in the citric acid cycle? Citrate synthase. That's the only one where it's the enzyme is describing the thing that it makes. So it's making citrate. Everything else, the enzyme is talking about the thing it started with. So like isocitrate dehydrogenase, it's taking a hydrogen off of isocitrate and making alpha-ketoglutarate. Alpha-ketoglutarate dehydrogenase, taking the enzyme off, making succinyl-CoA. How many molecules of CO2 are produced by the citric acid cycle?
2.
I need more than 3 voices.
2.
2. What enzymes?
The isocitrate dehydrogenase and alpha-K dehydrogenase.
Yeah, so the 2 in the bottom right-hand corner, right?
3 and 4.
So the isocitrate dehydrogenase, alpha-ketoglutarate dehydrogenase, those are both decarboxylation
Oxidative decarboxylation.
reactions, oxidative decarboxylation. Good. What inhibits pyruvate dehydrogenase complex?
NADH.
And why?
Because it's one of the things it forms.
Exactly, right? So our products inhibit, so we kind of have that feedback inhibition with our products, right? So ADP, that's going to make it go, right? Because we're trying to make energy overall. TPP is one of the 5 things on top of the reaction that's making it work.
Pyrophosphate.
And pyruvate is also on the left-hand side of the equation. So NADH is the only thing on the right-hand side is a product. So if we've got a lot of NADH, we don't necessarily need pyruvate dehydrogenase complex. So look for the products. The products are going to inhibit the enzyme. Things on the right are going to inhibit the enzyme. True, false. Citric acid cycle takes place in the mitochondria. Good. How many ATP does 1 molecule or 1 mole of FADH2 produce? 1.5. We got 2.5. Do we have any for 2 or 1? 1.5, right? So this one's 1.5. Gotta know it, gotta know. This is a guarantee question. So NADH, how many do we make in citric acid cycle?
3.
3. How many FADH do we make in citric acid cycle? 1, all right? So if we're making 3 NADH, we're making more. So that's how I remember. So it's worth more. 2.5, there's more of it, we get more out of it. FADH, we only make 1, so and it results in 1.5.
2.5 is for NADH. Yeah.
Gotta know it, gotta know it, gotta know it. Oh, missed that one. What's a definition of an antipyrotic reaction?
Used by other
The filler gap.
reactions.
So, so antibiotic reactions, they refill TCA cycle intermediates, so any one of the 7 molecules that have been extracted for biosynthesis of other molecules elsewhere in the body.
The other biomolecules like create the intermediates for the TCA. Is that how it goes then?
Yeah.
We were like discussing it yesterday.
So antibiotic reactions. So for example, we've got malate. So malate's used elsewhere in the body. So there's, I think maybe in the next chapter or the next couple chapters, we'll learn the malate aspartate shuttle. So that's helping to take things across membranes. So if we take out malate, the antibiotic reaction is going to produce malate and put it back into the TCA cycle to make sure that TCA cycle can work, the citric acid cycle can work. Because it's been removed, we got to replace it. But really what you're looking for is something that says TCA cycle, intermediate, biosynthesis of something else. If you see any of those 2 things, I don't think he's going to like confuse. He's not going to ask like, oh, it's taking out TCA cycle intermediates, or it's like putting in. He's not going to contrast it like that. So if you see intermediates and biosynthesis of elsewhere other pathways, that's going to be your answer.
There was like a quiz question that we had regarding it, and I don't remember the exact wording, but the answer was like all the above, and it was like fatty acids, something else, something else.
Oh, yeah.
What was that?
Yeah.
What was that?
Not sure what that question is.
Here's this quiz question.
He does. All right, next one. What are the coenzymes required for the production of acetyl-CoA?
Okay, I found it.
B, C, E.
All right, so what are the 5 from the top?
B, C, and E.
So we've got NAD, TPP, CoAsh, FAD, lipoate, right? So NAD, FAD, first 2 easy. All right, CoAsh, easy. We're making acetyl-CoA, so we need to have that CoA in there somewhere. And then just making sure you remember lipoate, TPP.
Is he gonna give us like trick questions like thymine, chloro, something like, and just spell them wrong? Because we'll just know it's just TPP and not actually know what.
He might, I mean, definitely know what it is. So let's see what it is. It's like this. Yeah, so it's thiamine pyrophosphate. I don't think he'll like misspell it intentionally. It might he won't like just switch a letter or something on you. It's gonna be pretty obvious, but it should just say TPP on the test. All right, you got the question?
Yeah, it was intermediates in the citric acid cycle are used as precursors in the biosynthesis of amino acids, nucleotides, fatty acids, sterols, or all of the above.
So it's all the above.
Yeah. So
What was the question again?
Intermediates in the citric acid cycle are used as precursors in the biosynthesis of and then A, amino acids, B, nucleotides, C, fatty acids, D, sterols, or E, all of the above.
yeah, so that one is just all the above. Yeah, it's just, I mean, like those are all roughly, I mean, if he didn't talk about something like that, then just, I don't know, I just go with all the above, typically. Especially if there's not like a and C on there. All right, what molecule does fumarate make? Is it fumarate?
No, malate.
Okay, so our gut instinct is to say, hey, fumarate, right? It's got the name in it. But remember, all of them except for citrate synthase, it's talking about the one before. So fumarate, enzyme fumarase makes malate, right? So answer is malate. What enzyme produces alpha-ketoglutarate? Isocitrate dehydrogenase, right? So
With the word alpha. Could you not find the symbol?
that was a lot of work. So, like I said, you just got to be able to take yourself out, say citric acid cycle, and then you're given a clue of, hey, isocitrate dehydrogenase, and just be able to put yourself back in there and know what each of those enzymes do individually. He can ask you anything about them. So what they make, or what enzyme makes something, or what they produce. So just know everything. You got to know everything about these cycles. There's a defect in the enzyme aconitase. What product will accumulate?
Citrate?
Left side of the room. What are we thinking? Why?
Because aconitase is for 2A and 2B, so then you're not going to have the unstable one, and then that makes isocitrate, and those are the 2 enzymes.
So aconitase is taking citrate and making isocitrate, right? So if we don't have that enzyme making isocitrate, we're just going to keep getting more citrate, just more and more citrate, because there's nothing turning it, making it into the other thing. So it's like if we're on an assembly line and we've got one worker putting on a door, we've got one worker putting on the hood of the car or what have you. Well, if you take out the worker putting the hood of the car on, so it's just going to accumulate. You're just going to get more hoods of cars, more hoods, and they're just going to accumulate as opposed to going and making the full truck.
So it was A? Yes.
Yeah. Or if y'all ever watched that Drake and Josh, where they're like working the assembly line at the sushi thing or whatever, and it's just like a nightmare. Anyway. All right. What reactants will inhibit pyruvate dehydrogenase complex? Anything else?
Nope, all those are coenzymes.
All right, so why NADH? Because it's a product, right? Everything else is a reactant. It's really simple if you know it. If you know the 5, it's really simple. There's going to be 3 or 4 easy points. What kind of reaction produces citrate?
Yeah. Yes. Condensation.
Condensation, right? All right, so we've got, let's just go through the order real quick. So of the types of reactions. So we've got condensation, and we're going to have a dehydration, rehydration, then oxidative decarboxylation, oxidative decarboxylation. Okay, and what enzyme is that for? What enzyme is that for? Synthetase, right? So synthetase has a T in it, so it's making the GTP. If there's GTP or ATP, it's substrate level phosphorylation. All right, what's the next enzyme? Or I'm sorry, type of reaction. Thank you. A dehydrogenation, and then we've got hydration, dehydrogenation, then condensation. We're back at the top, right? So we got condensation, dehydration, rehydration, oxidative decarboxylation twice. Then we've got substrate level phosphorylation. And we've got just a dehydrogenation, hydrogenation, rehydration. Was that hydration? And then another dehydrogenation.
Hydrate it and you dehydrate it again.
Exactly. So, got to know these types of reactions. Same thing. All right. What enzyme produces FADH2?
Succinate dehydrogenase. It sucks. It gets an F. Wait.
All right. So succinate dehydrogenase is going to make FADH2. So for those of you who weren't here last week, so you've got those two, you have the succinyl-CoA synthetase first, and then you've got succinate dehydrogenase, right? And it's kind of easy. One of those makes GTP, one of them makes FADH2. So both really important. Those are the only ones that make those things. So succinyl-CoA synthetase has that extra T in it, so it's going to make the GTP. The succinate dehydrogenase, it's a dehydrogenation reaction, so it's got to make an electron. It's got to take a hydrogen off and put it somewhere. And it would suck if you forgot that that's the only one that makes FADH2. So FADH2, succinate dehydrogenase. What enzyme produces GTP? We just said it. So this one's the succinyl-CoA synthetase. What enzyme is categorized as a dehydrogenation reaction? All right, so we've already gone through the order, right? So we just went through the order of all these types of reactions. All right, so is it on the right kind of side of the circle, or is it on the left side of the circle?
Dehydrate, hydrate, dehydrate.
It's on the left, right? So alpha-ketoglutarate, isocitrate dehydrogenase, those are going to be taking off the CO2s, right? So those are oxidative decarboxylation. But on the left, they're only making NADH, they're only making FADH2, so they're strictly dehydrogenation reactions. So the answer is C, succinate dehydrogenase. It could also be what enzyme?
Malate dehydrogenase.
Malate dehydrogenase, right? Because that makes NADH, right?
Yes.
Good. All right. Which of the following enzymes produces one molecule of carbon dioxide? A. Just jumps out at you, right? Isocitrate dehydrogenase. It's oxidative decarboxylation. It's on kind of the bottom right-hand side where both of those were, for those of you that are visual. Really simple. Or for those of you who aren't visual but you kind of remember the numbers, remember 1, 3, and 4 are our irreversibles. So, and we already said that 3 and 4 were irreversible because they're removing that CO2 from the equation. So lots of different ways to remember it. What reaction in the citric acid cycle are irreversible? Oh my God, I just said it. I'm just ruining everything. All right, scale of 1 to 10. How awesome is Andrew?
D. It's not up there. All the above.
Infinity. Yay.
21.
All right, so that's all the tests I made up for y'all. I think this test is going to be way more straightforward than the other one's going to be, just because what can he ask you from glycolysis or from citric acid cycle that I didn't just ask you now? You know the types of reactions, know what makes what produces what. Don't get confused by the order. So if you see what does fumarase make, you're not thinking, oh, fumarate, because it's in the name, right? You're thinking, you're going to just know it. It's going to be malate. Got to know which ones make NADH, which ones make FADH2. I've said this like 1,000 times now. Which one makes GTP, which ones are irreversible, and how feedback inhibition is going to cancel it. Is going to turn off or on enzymes. Same thing for glycolysis. Got to know which ones are irreversible, which ones produce NADH, which ones make and use ATP, which one, it's all the same stuff. What the types of reactions are, it's all the same stuff. Any other questions? All right. Get some sleep. Y'all feeling good? Y'all feeling good about it?
Yeah, but he doesn't.
I have a question. In terms of talking about magnesium and glycolysis,
Okay.
as a cofactor, what does MN stand for?